Hi, this is Dr. Bueller, and we're going to talk about our third section of antibiotics. In our first section, we talked about cell wall synthesis inhibitors, and in our second section, we talked about protein synthesis inhibitors. Here, we're going to talk about cell membrane, nucleic acid, and folate synthesis disruptors. For these, you're going to need to know the full mechanism of action, both the molecular and the cellular. And we're going to start with cell membrane disruptors. The cell membrane disruptors here consist of two polymixins and daptomycin. To understand the selective toxicity of these agents, you need to recognize that cell membranes from eukaryotes and prokaryotes are very different from each other. We generally think of cell membranes as just this phospholipid fluid mosaic that has a few proteins interlaid in it. But these cell membranes are actually far more complex. There are differences in the actual phospholipid components, the type of phospholipids, between eukaryotes and prokaryotes. There's different outer protein structures, there's different porins, there's different signaling and receptor components. Eukaryotes have a lot of cholesterol in their cell membranes, whereas prokaryotes don't. So there's these major differences that are going to allow us to target cell membrane disruptors to bacterial cell membranes without as much of an effect on ours. The second thing you need to think about is that the different cell membranes in bacteria also have different components. So in a gram-negative organism, you've got that outer lipid membrane and an inner lipid membrane, and those have somewhat different functions, so those are going to have different components. The gram-positive bacteria has its own cell membrane, which is different from either cell membrane in the gram-negative. So you can target drugs for the outer membrane of gram-negative, for the inner membrane of gram-negative, or for the membrane of gram-positive. So the first class of these cell membrane disruptors is polymixin. We have polymixin B and polymixin E. These guys insert into the inner cell membrane of gram-negative bacteria. So you've got the outer cell membrane, you've got the cell wall, and then the inner cell membrane. These polymixins are going to make their way through the outer and the cell wall into the inner cell membrane. These guys are not 100% selectively toxic, so they do have some action at human cell membranes. So when given systemically, they do have some toxicities. If you look at the structure here, the first thing you're going to notice is this outer head that is hydrophilic, and then this inner tail that is lipophilic. So when it comes up to the inner cell membrane, I'm going to draw that outer hydrophilic head on it here. The lipophilic tail is going to insert into the cell membrane, leaving that hydrophilic head out here. And then those are going to attach to each other, and they're going to form a pore. When these insert into the inner membrane, they're going to puncture holes inside the membrane into the cell, causing cell lysis. These are specific only to gram-negative, but they do have some toxicities systemically for humans. So we have polymixin B and polymixin E. Polymixin B is generally used topically, ophthalmically, or otically, so it's not going to have that systemic concentration, so you're not as worried about systemic toxicities. However, polymixin B does also have systemic routes, IM, IT, and IV, that are used for more serious infections. Polymixin E does include a nebulizer route, and that's important because it's one of the drugs that can be specifically used for cystic fibrosis. And in cystic fibrosis, that's a genetic disorder where the lungs make too much mucus, and you can commonly get bacterial lung infections. So having a nebulizer where you're actually inhaling the antibiotic can get that antibiotic right where it's needed. All of the polymixins, B and E, if given systemically, so remember not if given topically, are going to have a risk for nephrotoxicity, so that's renal toxicity, and that's a fairly high percentage of patients, as well as neurotoxicity. Because of that, in the systemic routes of administration, we've got boxed warnings for polymixin B. Got two of them here. One is for IV administration for nephrotoxicity or neurotox, and the other for polymixin B in any type of systemic administration to only be given to hospitalized patients. So the second class of cell membrane disruptors includes daptomycin. And if you look at the structure of daptomycin and you think back at what we just saw, you're going to see a huge similarity here. You're going to see an outer head that is hydrophilic and an inner tail that is lipophilic. So it's basically doing the exact same thing as the polymixins, except it's targeting a different cell membrane. So this is targeting gram-positive cell membranes. It's doing that same thing in terms of inserting that lipophilic tail into the cell membrane, and then the hydrophilic heads are going to bind together, forming a pore, puncturing that hole in the cell and causing cell lysis. Remember that bacteria have different components for the gram-positive cell membrane and the two different gram-negative cell membranes. So this drug can target components of the gram-positive cell membrane. Stapdomicin is then specific for gram-positive. It also can't permeate the outer membrane of gram-negatives. It does have some specific adverse effects. It can induce rhabdomyolysis. We often just refer to that as rhabdo. And because of that, patients are often going to be given monitoring for creatinine and phosphokinase levels. Concomitant use of statins are avoided because they also increase risk of rhabdo, so you don't want to have two drugs at the same time increasing that risk. The next drug class are the nucleic acid synthesis disruptors, and we're going to talk about DNA gyrase inhibitors, RNA polymerase inhibitors, and the reactive species agents such as metronidazole and nitrofurantoin. So here with the nucleic acids, so here with the mRNA polymerase inhibitors, those include rifamycins. The topoisomerase or DNA gyrase inhibitors include the quinolones, specifically fluoroquinolones, and these reactive species generators, metronidazole and nitrofurantoin. The rifamycins include rifampin and rifaximin, and rifaximin is just a PO unabsorbable form of rifampin. So remember, when we talk about PO, all that means is you're taking orally, so you're taking by mouth and swallowing it. That doesn't tell you whether it is absorbed by your GI or not. So when we talk about orally bioavailable, that's telling you about whether it's absorbed by your GI. So rifampin is taken PO and is orally bioavailable. Rifaximin is taken PO but is not orally bioavailable, so it's going to stay in your GI tract. The rifamycins inhibit bacterial RNA polymerase. Bacterial RNA polymerases are structurally somewhat different than ours, so that's where we're going to get our selective toxicity. And RNA polymerase, you remember, is just what we use to transcribe DNA to mRNA. And so if we block that RNA pol, then we don't make messenger RNA. The gram specificity for this is mostly gram-positive, but a very important point of this is it also does target mycobacteria. And tuberculosis is one of the main things that we're concerned about there, because there's not a whole lot of drugs that are active against tuberculosis. The rifamycins have some interesting properties. One of them is that you need to avoid alcohol, and that's not due to a reaction between the drug and the alcohol, but rather due to the risk of cumulative liver damage. For some reason, these can change the color of your body fluids, so your sweat, your urine, and even your tears can begin to turn somewhat yellow or orange. And rifampin, which again was absorbed, undergoes biliary elimination, and because of that can have enterohepatic circulation. So again, it's eliminated within the bile, that goes into the GI tract, and then it's taken back up lower down in the GI tract and reabsorbed into the body. So that's going to increase duration of action. Rifaximin, we said, is the same thing as rifampin, but it's not orally bioavailable. So when taken PO, it's just going to stay in the GI tract, have fecal elimination, and no concerns about metabolism. Rifampin itself, some of the concerns we have about it are it's a strong inducer for CYP3A4, so that's going to cause the potential for drug interactions with any other drugs that are metabolized by CYP3A4, and it can cause both hepatotoxicity and renal toxicity. So it's recommended to have liver function tests as well as creatinine level tests. The reason these aren't a concern with rifaximin is that rifaximin is not going systemic. The second class of nucleic acid synthesis disruptors are one of my absolute favorite antibiotic classes. I do have a favorite. This antibiotic class just fills me with joy, and these are the fluoroquinolones. So they're a quinolone backbone, and they have a fluoride group on them. The drug names here are ciprofloxacin, moxifloxacin, and levofloxacin, and there's also a new one, delafloxacin, but it's not in common use yet. You can remember these drugs are fluoroquinolones because they have the fluo in the name. The mechanism of action of these is to inhibit bacterial DNA gyrase. Now, DNA gyrase is a form of a topoisomerase. So topoisomerases are enzymes that break down DNA supercoils. So if you're old enough to remember the horrible frustration of phone cords, the cord itself had a coil to it, and then if that coil became too tight, it would coil back onto itself in a supercoil. Now, DNA does that too. That's beneficial for DNA because it helps compact it so it doesn't take up as much space. But if you want to go in and transcribe into mRNA or duplicate the DNA for cell replication, you're going to have to open that up so the replication enzymes can get in there. So the first thing you're going to need to do is break apart that supercoil and unwind that. That's the function of topoisomerases. This bacterial DNA gyrase is just one form of topoisomerase. So when we inhibit that, we're preventing the relaxation of supercoiled DNA, and because of that, we can't replicate DNA and we can't produce mRNA. And again, I remember that these drug names are in the fluoroquinolone class because of flow, but I also remember the mechanism because of phlox. And I think of dental floss. And if you twist dental floss in your hands and you twist it tight enough, it'll supercoil back onto itself. So don't ever forget, ciprofloxacin is a fluoroquinolone, and because it's a phlox, it acts on supercoils. These guys are broad spectrum, and they have a couple of fairly unique properties. One is they can cause photosensitivity, and there's two types of photosensitivity. So you can have phototoxicity, and you can have photoallergy. And these are two completely different mechanisms. So phototoxicity occurs when the drug is actually converted by UV light into reactive species that are going to attack whatever molecules are around them. And that's really similar to what's happening when you get a sunburn. So drugs with phototoxicity are going to increase sunburn type effects when exposed to UV light. Drugs with photoallergy are even more interesting because obviously you can't be allergic to light. But what's happening here is when these drugs are exposed to UV light, they're altered in a way that makes them allergenic. So it makes them an antigen. When they're made an antigen, then T cells are going to come and attack that antigen. So that's an actual true allergic response. And similar to other T cell responses, you can get a rash. So that's different from the sunburn type effect. Fluoroquinolones actually have both, and so usually people are told to stay out of the sun or use sunscreen. These are also what we're calling a ditri, and ditris were agents that chelated di or trivalent cations. And so those cations are chelated in here. For the fluoroquinolones, that means that you avoid taking it orally with dairy products or calcium supplements, iron, magnesium, or aluminum containing supplements or antacids. Those will chelate the fluoroquinolone inside the stomach and it'll reduce the absorption. So the problem here, the reason you don't take them together is because it reduces absorption of the antibiotic. Fluoroquinolones can have some nasty side effects. When we thought back on tetracyclines, which were also ditris, those accumulated in growing bone and tooth. The fluoroquinolones do a similar thing. They accumulate in bone, but they also accumulate highly in tendon and cartilage. So these are avoided in pregnancy to avoid damage to that growing cartilage in the fetus. And they also have a boxed warning for adults that it can accumulate into adult tendons and cause inflammation and, in the worst case scenario, cause rupture of the tendon. These can also cause QT prolongation, so torsade de poids. These can also have seizure and neurotoxic effects, and that seizure is because these drugs are also very low affinity antagonists of GABA A receptors.